Every physical function your body executes right now is directed by a chemical manual. To find it, look deep within the cellular level, where an entire organism's blueprint is housed inside a microscopic nucleus. This 3D DNA model resembles a twisted ladder, with outer rails formed by a continuous sugar-phosphate backbone. Zooming in, the rungs are nitrogenous bases following strict pairing rules. Adenine always pairs with thymine, and cytosine always pairs with guanine. Just as arranging distinct letters creates words and sentences, the specific linear sequence of these AT and CG base pairs acts as a literal, readable code along the DNA molecule. The English language uses 26 letters to construct its entire vocabulary. The instructions required to build and operate a complete, highly complex human body rely on an alphabet of just 4 chemical bases. DNA operates as a highly efficient information storage system. The exact sequence of its chemical rungs constitutes the foundational language written inside every living thing. If you extracted the DNA contained within a single human cell and stretched it end to end, the thread would measure roughly 2 meters long. Storing that volume of material inside a microscopic nucleus creates a severe spatial problem. To fit inside the nucleus without becoming a tangled mess, the loose threads of DNA, known as chromatin, tightly coil and fold themselves into distinct, rod-shaped structures called chromosomes right before a cell divides. This graphic of a condensed chromosome shows distinct bands illuminating along its arms. These localized segments along the packed thread are genes, the specific functional units of the DNA code. This extreme dynamic compression allows the cell to safely store, organize, and inherit the massive library of genetic instructions required to operate the human body. But holding the instructions is only half the process. DNA is safely trapped within the nucleus as a locked reference book. It does not assemble any physical structures directly. This animation illustrates the first step of execution, transcription. The DNA double helix temporarily unzips, allowing the cell to copy the exposed code into a mobile messenger molecule called mRNA. Once the copy is synthesized, the mRNA leaves the nucleus and migrates out into the cytoplasm to seek out the cell's active manufacturing centers, which are called ribosomes. Here we see the second step, translation. As the mRNA feeds linearly through the ribosome, the complex reads the code to assemble a precise chain of amino acids, which fold into a specific 3-dimensional shape. Without this execution phase, the genetic code is entirely inert. Transcription and translation are the mechanical bridges that convert dormant genetic data into functional physical proteins. Once assembled, these proteins leave the cell's manufacturing centers to act as the actual physical workers of the human body. They dictate the macroscopic functions you rely on every day. Proteins synthesize enzymes to regulate your metabolism, facilitate the growth of dividing cells, and physically structure your internal organs. These specific protein expressions are also responsible for Mendelian traits. They dictate the visible characteristics we recognize, variables like eye color, hair texture, and height, first tracked by researchers like Gregor Mendel. Because the cellular machinery copies and reads the original DNA sequence with intense precision, these biological characteristics pass reliably from one generation to the next. The macroscopic reality of the human body relies entirely on microscopic assembly.